Hello class, in today's lesson we're going to be looking at the three central processes that go along with DNA and RNA, which is replication, transcription, and translation. Each one of these is a vital process to our cells and either makes more copies of our DNA or makes it into a usable form that our cells can use to make our proteins. So the first one that we're going to start off with is DNA replication. And what it is a process where DNA makes an identical copy of itself. There's a couple of steps that we'll go through that takes it along there, but that's essentially what replication is. We've covered this vocabulary word in the last chapter of replication. When it came to learning about mitosis, it's one of the things that goes on during interphase. But DNA replication is a process where DNA makes an identical copy of itself, one-to-one. So if we take these Lego bricks, which I'll be using later on, it'd be like having two yellow bricks. The first one was its first set. After replication, it makes a second brick. So here's how it works, and it's going to run down in a series of steps. The very first step is that the DNA has to unwind itself. The other day we talked about DNA being in a twisted ladder formation. The first thing that we have to do is untwist the DNA. So the DNA is all twisted. It's got its ladder shape. What we have to do is untwist it so that we get a more normal ladder shape. So the first thing that happens is unwinding. And this is all done by enzymes, which are those chemicals, those proteins in our body that speed up reaction. The second step is that once we have the DNA lined up like a ladder, we have to unzip the DNA connections. We talked about the base pairing rule, where A's go with T's and G's go with C's, adenine with thymine, guanine with cytosine. And that's what we're going to do. We're going to break those bonds apart from each other. So it's got to unzip, just like a zipper. It unzips itself. When the unzipped end are going to be our free nitrogen bases, to which new nitrogen bases can come in and pair up with. So what ends up happening is that we get on the sides, I'll use my Lego bricks for an example. Let's say on one side we got blue here. Let's call blue adenine in this example. After it unzips and unwinds, this adenine will want to combine with a thymine. Let's have red be the thymine. So what's going to happen is this adenine is going to be just sitting there until a thymine comes in and joins up with it. And now we have our double-sided DNA once again. And that's what the third step is that the free nucleotides come in and pair up. So they pair up at first. After they pair up, the bond is going to form. That's like pushing the Legos together. Because right now, the bond's fairly weak. It can fall apart. We don't want that to happen to our DNA. So what we need to make sure that happens is that the bond forms solidly. And that's when you would press two Lego bricks together. So the bonds form. And at the end, what we end up having done is that we get two new strands. After all the bonds are reformed, we get one strand that is completely new, and one strand is the old strand, the template strand. And what happens is that after they reform, they unzip, they come back together, or one side becomes completely new, and then they will rewind themselves back up into their double helix. Now, along with replication, the question goes on with, why do we replicate? Our DNA needs to be replicated because of mitosis. Each new cell needs to have the same information as the previous cell, so that's where DNA replication comes into play. Without DNA replication, we wouldn't have the same instructions in all of our cells, which means our cells would lose some of their functions, which in that case our cells will die, which if our cells die, then stuff wouldn't be good for us. In the second part of today's lesson, we're going to be talking about the process of transcription. Now, transcription is a little bit different. Transcription is going from DNA and converting it into RNA. Now, why do we need this process? This process is useful because the ribosomes themselves can only use RNA to make proteins. So we need to convert the DNA into RNA in order for the ribosomes to use them as proteins. Now, what's the difference between DNA and RNA? We already went over that in class the other day. DNA is double-stranded, whereas RNA is single-stranded. one of the bases changes. So in DNA we have thymine, which I believe we represented with a red brick earlier in our lesson. Whereas in RNA we have uracil, which in this example I'll represent with this white little brick. Now, one of the differences again with DNA and RNA is the type of sugar that they have, and structurally they are different. They are built differently because of either the deoxyribose sugar or the ribose sugar. Now, for that, I'm going to represent that using two different types of Lego bricks. It would be like using our rectangular Lego brick when we are referring to DNA. Whereas when we are referring to RNA, it would be in a slightly different form. So I'd use the square bricks in this case. Now, what happens during transcription is very similar to what happens during replication. And I'll kind of outline it here using our Legos. So we have our, let's call this a section of our DNA molecule. We got our adenine, which is going to be represented by our blue, and we got our thymine, which is represented by red. Right now it's DNA. It's double-stranded, two-sided there. What's going to happen is that the DNA is going to unwind and unzip. So just like before, we got the unwinding and the unzipping of the DNA. So in a sense, the bonds are going to be broken. And in this case, we're only going to look at one side of the DNA. In this case, I'm choosing the one that has adenine to it. Now, what is going to happen is that an enzyme is going to bring in RNA nitrogen bases to pair up with the strand of DNA, and only one side of the DNA. So in this case, for DNA, we have an adenine. The complementary base for an adenine is going to be a uracil, which I'm representing using the white brick. So those two would go together. The square would represent the RNA, the rectangle shaped would represent the DNA. Now when it all is said and done, what happens is that all of the RNA that gets produced will go into making a gigantic long chain that would be forming the DNA basis. So it would look something like this. This would be our mRNA strand. It would be a combination of all of the nitrogen bases lined up in a row. Notice that in this case there's nothing on top or on bottom, it's all just one line. That is because RNA is single-stranded. So this would be the message that would carry on to code for a protein in this case. Our last process is the process that goes into forming a protein. This process is known as translation. Now there's going to be some confusion between transcription and translation because the words just sound familiar there. But think about this in terms of our DNA and RNA into our proteins. Both DNA and RNA are both nucleic acids, so they look very similar to each other. So think about like a foreign language. When you transcribe something, you're taking that language and putting it from like a verbal tense from me speaking, and you're writing it down on paper. That'd be like transcribing it. You're still using the same language, it's just in a different format. One is verbal, the other is written. For translation, this would be like moving from English to Spanish. It's got the same code to it, but it's in a completely different language, so that it's just going to look different completely. And that's what happens during the translation process. We go from moving from a nucleic acid to forming a protein in it. So what is translation in general? Essentially, the process of translation is where an mRNA strand, so that's messenger RNA, is read. And it's read by a ribosome. And what happens is that ribosome then uses the mRNA as a code to then form a protein. And each mRNA would code for a different protein sequence. And that protein sequence would create its own individual protein, which would then go on to carry out its own individual function. Now, how this is read is that the ribosome recognizes the mRNA in groups of 3. And we call these groups of 3 a codon. So I've prepared some of our Lego samples as codons. So we've used in the past, we've used the little square bricks as our RNA sequences. So here I've taken 3 of the bricks and made a little codon out of them. 3 consecutive mRNA bases. And I got a couple of them here for sampling processes. Now notice that there can be 2 of these same types of nucleic acids or nitrogen bases lined up right next to each other. So there could be 2 uracils right in a row there. But it doesn't always have to work that way. Now, what is going to happen is that with these 3 consecutive mRNA bases, each one of them is going to code for a specific amino acid. Now, an amino acid, as we learned about last chapter as one of our vocab words, is what goes into making up a protein. And there's 20 of these amino acids. Now, I know I don't give you a spot to write that down, but somewhere in the corner, close to, or somewhere on that page, put down that there are 20 amino acids, because that's going to become important when we're doing our next step here. So our codon, 3 consecutive mRNA bases that code for a specific amino acid. Now, when we are using this, the ribosomes don't know this. The ribosomes can read this automatically based on its chemical signature. But for us as scientists, we have devised a little kind of like a decryptor or like a code to break the mRNA base code. And that's what's called a codon chart, where it has every single codon, which is 3 consecutive mRNA bases, and it tells you what specific amino acid those 3 bases in a row will code for. And that's how we're able to figure out how different proteins are made. And the steps for how the translation process works are as follows, because it's a lot different from how replication and transcription work, because it's going from into a completely different language. So what you have is the mRNA. So we can take the full sample of our mRNA here. So our full entire mRNA is going to attach itself to a ribosome. And as it's attached itself to the ribosome, it reads the entire mRNA code, just like you would read a book. It starts, it goes from left to right, and it reads all the bases. And it's looking for a specific start sequence, what's called the start codon. Once it finds that start sequence, it starts building its protein. In our case, and what's been found to happen all the time is that the start codon for any type of protein is coded by adenine, uracil, guanine, in that combination, or AUG, AUG. And that's going to code for the amino acid methionine. So what happens is that the mRNA is read all the way up until that point to where it starts to build the protein. So this is where all the actions happens. So we go on to step 2. So we got our AUG. This is going to start our starting our building of our protein. What's going to happen is that our tRNA, which I'm using these gray little bricks to represent our tRNA, is going to bring an amino acid to connect to the ribosome. So it brings in an amino acid. We're going to call this part the amino acid right now. So it brings in its first part. Now what's going to happen is that it's going to read the next 3 bases. So the next 3 bases come into play. And the next 3 bases bring in a new tRNA into the equation. Now what happens at this point is that the new tRNA kicks the old tRNA out of its place. But before it does that, it will take its amino acid from it. And that tRNA is free to go and grab another amino acid that's just floating out there in the cytoplasm. So right now we got a growing amino acid chain here. Now new amino acids are continuously bonding to the previous ones, creating a protein. So the old ones are connected to the newer ones. And it keeps on building and building and building and building and building and building until it reaches what we call a stop codon. Now there's 3 different stop codons, and we'll talk about what these are in class. But there's 3 different stop codons. Now these don't code for a amino acid. These are not an amino acid whatsoever. What happens essentially is that, let's say the next base is our stop codon here. What happens is that a tRNA comes into it, binds to it, but what happens is that there is no amino acid connected to there. So when it goes to try to connect to the newer amino acid, there's nothing left to connect to, so then we have our completed protein here. And this protein can then be packaged off and do whatever it is that this protein needs to do. Now, obviously, why does this process exist? All these processes exist to make all of our proteins in our body.